Hello, this is Dr. Christine O'Connor, and today, as part of the Introduction to Medicinal Chemistry, I'm going to talk about bioinorganic chemistry of S-block metals. So if we look at the bioinorganic chemistry of S-block metals, the S-block metals we're talking about are the alkali metals, which are in group 1 of the periodic table, and the alkaline earth metals, which are group 2 of the periodic table. So the bulk quantities of group 1 and group 2 metals are required by most organisms, mainly to balance the electrical charges associated with negatively charged organic macromolecules in cells, and also to maintain the pressure inside the cell to keep it turgid or rigid and prevent it collapsing. So macromolecule just means a big molecule. In spite of the close similarity of chemical properties between sodium cations and potassium cations, and between calcium and magnesium cations, their biological functions are very different. So magnesium and potassium are associated with intracellular activity, so what's going on inside the cell, whereas calcium and sodium are associated with extracellular activity, or what goes on outside of the cell. So the two sets of ions may not replace each other. So again, just reminding ourselves of group 1 here in the periodic table and group 2 here in the periodic table, and we're looking at these cations here of the various elements. So magnesium 2+, so a magnesium 2+ forms a complex with ATP and is a constituent of phosphohydrolases and phosphotransferases, which are enzymes for reactions involving ATP and energy release. So magnesium is also essential for the transmission of impulses along nerve fibers and is a component of chlorophyll. Potassium then is essential for the metabolism of glucose inside the cell and the activation of some enzymes. And calcium is important in blood clotting and is needed to trigger the contraction of muscles and to maintain the regular beating of the heart. So calcium is also used outside the cell as a structural factor in bones and teeth as apatite, and the enamel on teeth as fluoroapatite. So each of these cations have their roles within biological systems. And sodium, so the different ratios of sodium potassium inside and outside the cell produces an electrical potential across the membrane, which is essential for the functioning of nerves and muscles. So the movement of glucose into cells is associated with sodium, and they enter the cell together, and this is favored by a high concentration gradient. So distribution of sodium, potassium, magnesium and calcium, the concentration of these ions varies systematically. So an example, if you analyze the fluids inside animal cells, you'll see that potassium is 0.15 molar, which is moles per liter, whereas sodium concentration is 0.01 molar, which is moles per liter. Outside of the animal cells paints a different picture. So for example, in the blood, potassium has a much lower concentration of 0.003 molar, and the sodium cation has 0.15 molar, so it's a much higher concentration. So these ions must pass through a membrane that separates the inside of the cell from its external environment, and the membrane is extremely selective, so only certain molecules can pass through, and in some cases molecules can only pass in one direction. So this process of moving ions through the membrane is known as pumping. So some of you may be already familiar with the sodium ion pump. So the simplest concept is that of a carrier in the membrane which can combine with the ion. It carries it to the other side and then releases it. So in this case, the carrier is a phosphoprotein, and this consists of a phosphate group with the protein serine, and serine is shown in this diagram here. So there's different types of sodium phosphates. You have mono, which is depicted here, sodium monophosphate, so there's one phosphorus group here. Sodium diphosphate, so again there's two phosphates bridged by an oxygen, and sodium triphosphate, so you have 1, 2, 3 phosphates, and again bridged by oxygens. So the phosphoprotein, or we're representing it by the P, binds the potassium ion at the outer wall and carries it to the inner wall. Then you get phosphorylation occurring, which is the phosphoprotein phosphatase, which is represented by PP, and is formed, and the potassium is then released. So the phosphoprotein phosphatase then binds with sodium at the inner wall and carries it to the outer wall. Dephosphorylation then occurs, returning to the phosphoprotein, and the sodium is then released. So the phosphate group needed for phosphorylation is obtained then from ATP. The selectivity of these phosphoproteins is due to the size of the cation. So sodium cation is smaller than the potassium cation. So controlling calcium ions, so the calcium 2 plus ion concentration is controlled by two hormones, calcitonin and parathyroid, and calcitonin inhibits the release of calcium while parathyroid mobilizes calcium from the bones. So bone structures are combined of solid octocalcium phosphate on a framework of the protein collagen. So the calcium salt must be insoluble, otherwise it will be washed out of the bones. So in order to absorb calcium from food, it must be present as a soluble salt. In muscles then, each muscle fibre is made up of thousands of fibrils, and the proteins myosin and actin are organized in the fibrils in the form of two types of filaments, one thick and one thin. So the thick filaments are made up of myosin molecules, which are long thin molecules, about 150 nanometers. So remember, a nanometer is 10 to the minus 9 of a meter, with a thicker head of 20 nanometers. The head, which is 4 nanometers, is about twice the thickness of the thin portion. The thin filaments are made up of doubly stranded chains of actin molecules, which are globular molecules, about 5.5 nanometers in diameter. And stretching of muscles occurs because of the presence of negatively charged groups on myosin and actin, which repel each other. Contraction of a muscle is brought about by the calcium 2 plus, which is able to form a bridge between those negative groups on the actin and myosin. So without the calcium 2 plus ions in our muscles, the muscle would not contract. So this is just a diagram showing the role of calcium and myosin in muscle contraction.